Okay, so today I will show you how to do the Sds-Page and Western blotting experiment. Okay, so because the first step of the Western blotting is to separate the proteins based on their molecular weight. Okay, so which means we will first do the Sds-Page experiment. Okay, so this is all we need for today's experiment. Okay, this is the precast gel we bought from the BioRed, and sample loading buffer 4 times concentrated, and the beta ME. Okay, and for the sample we picked station 13. Okay, the reason to pick a station 13 for the money group is because the positive and the negative transfected sample have very similar protein concentration. Okay, so we don't need to adjust the protein concentration. Okay, so and also what we need is the gel running apparatus. Okay. So this gel running apparatus allows us to run 2 gels at the same time. Okay, there are 2 inserts. Okay, here and here. Okay, but we don't need. We only need to run 1 gel. Okay. So this is all we need for today's experiment. Of course, we need pipette tips and pipettes. Okay. So what we are going to do first is we wanted to, I'm going to transfer, and we will follow the protocol in the lab handout. Okay, in from page 28 to page 32. Okay. So because we don't need to adjust the protein concentration, so we directly move to step 3. Okay. Okay, so we are going to mix 20 microliter beta ME and 4 times concentrated sample buffer. Okay, which means we will combine 90 microliter of concentrated sample loading buffer. Okay, this is a colored sample loading buffer. Okay, and 10 microliter beta ME. Okay, to prepare the sample loading buffer. Okay, because beta ME is a reducing agent, it's not very stable, so we add just before we wanted to prepare the protein samples. Okay, and then I'm going to transfer 60 microliter of positive and negative sample to the 2 tubes. Okay, positive one. And a negative. Okay, and then we are going to add 20 microliter of the thermal-loading buffer we just prepared to each tube. Okay, so the purpose of thermal-loading buffer is to completely denature protein and to form the linear structure. In order to do that, we also wanted to heat the protein at 95 degrees Celsius for 5 or 10 minutes, for 5 minutes. Okay, we will do this in the air bath. Okay, which is already set at 95 degrees Celsius. Okay, so we are going to put it inside and wait for 5. We can vortex a little bit. Okay, in 5 minutes. And during the thermal heating, we can set up the gel running apparatus, gel running system. Okay, so what we are going to do is we wanted to take out the precast gel from the package. Okay. Okay, and also don't forget to remove the tape before use. Okay, you are going to remove it. Okay, and what you are going to do, you wanted to insert the gel, precast gel, into the insert here. This is the gel running apparatus you wanted to insert. There is only one direction you are able to insert, so let's try this one. Okay, it's good. If you do it in the other way, it won't fit. It won't completely get down. Okay, there's something on the top. Okay, so this is the right way to do it. Okay, and this gel, there are total 18 wells. Okay, which means that we are able to maximally load 18 protein samples. Okay, so the next step we wanted to do is to add sample loading buffer, sample running buffer. Okay, so this is 1 times Sds-Page gel running buffer. Okay, there are many bubbles. This is because we added SDS inside. Okay, what we are going to do, we wanted to pour. Okay, here I also wanted to mention this is the cathode side, and the bottom will be the anode side. Okay, because the Sds-Page will move the protein from the cathode side to the anode side. Okay, so what we are going to do, there is a chamber on the cathode side to hold the sample running buffer. Okay, so what we are going to do, we wanted to pour sample Sds-Page running buffer into the cathode side. How much to pour? You wanted to pour to the full amount. Okay, and for the anode side, you just needed to pour maybe 1/4 of the height. Okay, this is sufficient. Of course, you can pour more. Okay, but this is sufficient because it already covered the cathode side, electric wire. Okay. Okay, we don't need to use too much sample Sds-Page running buffer. Okay, what we are going to do next is we wanted to remove the comb. Okay, and you wanted to remove vertically. Okay, in order to show the sample loading buffer. Okay, what we are going to do, you grab the comb and pull up. Okay, so now you can see there are 18 sample loading wheels labeled with the number from number 1 to number 18. Okay. Okay, so this is the sample after heating at 95 degrees Celsius for 5 minutes. Okay, it's a light blue colored sample. Okay, and you can imagine now the proteins formed the linear structure. Okay, not a 3D native structure anymore. I also wanted to mention when we do the Sds-Page Western blotting, we need to use pre-stand marker. Okay, pre-stand protein ladder. Okay. Okay, so what we are going to do next, we wanted to load the 3 samples in positive, in negative transfected sample, positive transfected sample in a pre-stand marker in this order. Okay, we wanted to load in triplicates. Okay, why we wanted to do triplicate loading? This is because the first set will be used to stain Coomassie dye in order to reveal the abundantly expressed protein bands. Okay, and the second set we are going to blot with anti-EGFP antibody. Okay, for the Western blotting analysis of EGFP expression. And the third set will be used to blot with anti-beta-actin antibody. Okay, to analyze beta-actin expression. Okay, and for the Western blotting analysis, and the beta-actin analysis expression will serve as some housekeeping gene expression, which should be the same between your negative transfected and positive transfected samples. Okay, we will use that as a control. Okay, when we analyze EGFP protein expression. Okay, so usually when you do Western blotting, you need to analyze a housekeeping gene expression, which can be beta-actin or some GFDH or some other genes. Okay, so that's why we wanted to load the 3 samples in triplicates. Okay, and we will load from well 1, okay, till well 9. Okay, so how to load the sample? We will use the gel loading pipette tip. Okay, and this tip, you can see it's taller. Okay, the box is much taller than the tip box we used. Okay, so you can see the difference. We can compare, okay, briefly to see the difference. And the gel loading tip is much longer, okay, as compared to this is the 10 to 20 microliter pipette tip. Okay, much longer, and the front of the tip, okay, is a little bit thinner. Okay, so which means you are able to insert into the well, deep of the well, to load your sample. Okay, so we will use this long tip to load the samples. Okay, how much to load? We will load 15 microliter of the protein samples and 8 microliter of the pre-stand marker. Okay. Okay, we will load the negative sample first. Okay, what we are going to do, you want to insert the front tip to the bottom of the first wheel, okay, and slightly and gradually pipette the protein sample out. Okay, so I think one wheel, the maximum volume you may be able to load probably maybe 30 microliter. And now I loaded 15 microliter. You can see it's about the halfway to the top of the wheel. Okay, so which means the maximum loading volume is around 30 microliter. Next one, I'm going to load the positive transfected sample to wheel 2. Okay, and then I'm going to adjust to 8 microliter. Because pre-stained marker is highly concentrated, you don't need to load that much. 8 microliter is sufficient to see the protein bands. Okay, in wheel 3, it will be the pre-stained marker. Okay. And we will repeat the loading, okay, for the other 2 sets. Okay, so now we are done with the sample loading. Okay, so next we are going to connect the power cord, the power supply. Okay, and I first put on the cover. Okay, here, don't forget, black to black, red to red, okay? Okay, and then this is our power supply, black to black, red to red. Okay, set wattage current. Start. For 60 to 90 minutes. Okay.